Just check.
And then you can close microphones.
Oh, yes, I can. Okay, but then I have to go out from my screen
No,
again.
no, no, you're okay. You're
Okay.
absolutely okay.
Mm,
Okay, you're
Okay.
absolutely okay.
yeah, I think we are live now. So, okay, then we can start. Welcome, Christine.
Okay, good morning, everybody. Our community from the Biomedical Society chapter Puebla, or Puebla chapter. Today is a great pleasure to have a very nice guest today from the University of Heidelberg. Let me introduce her or give a couple of words about her. She is Christine Selhuber-Unkel. She is now professor for molecular systems engineering in Heidelberg University. But before that, she made her master's in physics in Uppsala University, her PhD in the University of Heidelberg, actually, with some postdocs in different institutions. For example, one in the Department of Biophysical Chemistry at the University of Heidelberg, um, also a postdoc in the Niels Bohr Institute in the University of Copenhagen in Denmark, and also in the Zoological Institute in Kiel. And there in Kiel, actually, from there we know each other. She was a professor from 2011 to 2020. She was the professor for the biocompatible nanomaterials in Kiel, and she has several awards. For example, one from 2008 was the Otto Hahn Medal of the Max Planck Society, and also a couple of research grants from ERC, first for starting grants and consolidator grants, also I think for the professorship. So, and with several papers on the area of biomedical stuff. So it's a great pleasure to have you here, Christine. Yeah, with no more words now. Yeah, you can start. Thank you for being here in Mexico.
Yeah, thanks a lot, Enrique, for inviting me and for
Yeah,
the nice introduction. So it's amazing how small the world can become. So, I mean, we met in Kiel, then we met in Mexico, and now we are online. So at least it works. It's perfect. Thank you.
great. Yeah.
So I will try to introduce into smart materials for controlling biological systems, and I will give you in the end 3 types of views on such systems. So before I start, I just want to show a bit of what my group is doing and what we are doing in Heidelberg. So I'm a physicist by training, which I think explains a lot of the methodology that we are using and a lot of the thinking that we are having. And we work at the interface of biology and chemistry, material science, and investigate such systems with methods from physics. So a lot in our group deals with how to control cells by materials, by 2D and 3D structures, also 3D printing, by functionalization. We also have biomimetic systems, and we are also using biohybrid systems. So, I mean, Heidelberg is a really famous place for doing physics. Unfortunately, the view from our department and our institute is not that nice. So this is the view from the theoretical physics department. They are sitting in a place called, or in a street called Philosopher's Way, and it's really amazing. They have their very old traditional houses, and they have this view in the end here. So we are sitting on a different campus where all the life sciences and natural sciences and also the medical departments are clustered. So we are really all on one spot. and it's pretty efficient to collaborate with other groups. So the institute that we are working at is the IMSI, the Institute for Molecular Systems Engineering, and we are still waiting for the finalization of our building, which is located between the Center for Advanced Materials, where we have a big clean room facility, and also the European Institute for Neuromorphic Computing. So it's a perfect place, really being embedded between material science and other institutes on campus. We are also strongly supported in Germany through initiative called Excellence University Initiative. So there are funding lines where they try to find the sort of most excellent strategies of the universities, and Heidelberg was awarded with this grant a few years ago. And there is a so-called flagship initiative where we can really have some startup funding and initiate new projects in the field. And the three subjects that I want to talk about today, these are photoresponsive biomaterials, microstructured biomaterials, and microengineered actuators. I mean, photoresponsive biomaterials is a part where we try to really use the material properties to control cells, but we not only use material properties and mechanics, we also use structures. This will be the second part of my talk. And then I will just have an excursion to a field that has less to do with biomaterials, but I think will be very interesting for you and might also grasp the interest of those listeners who are less interested in biomaterials, but more in robotics or classical composite materials. So why photoresponsive materials? I mean, responsive materials in general are very interesting. I mean, this is just an example from literature. I mean, a bullet is hitting into this window, and it is self-healing in the end because there is a reactive liquid, and the window can heal out in the end again, just by the interaction of the liquid and the oxygen that comes in when the bullet hits this window. So responsive materials are, of course, interesting for having sustainable systems, and you can really change the materials dynamically. Also in biomaterials, responsive molecules are really interesting. So this is an example where a group used a functional surface, a glass slide, and they had polyethylene glycol molecules on top, and the cells were not able to attach to them. So this happened here in this ring, in the outside of the ring and in the center of the ring. So here you can see some cells. That was an area where the cells were allowed to attach, but all the area around was just covered with these polyethylene glycol molecules. But these polyethylene glycol molecules, they were connected to the surface through photocleavable linkers. And when they now shone light, UV light, onto the sample, then the PEG was released and the cells started to adhere. And this is now what you will see in this video here. So the cells are initially just adhering in this ring here. And then I start with the UV flood exposure, and the PEG goes off, and the cells can explore the full space around this ring. So this is, of course, just a unidirectional thing. I mean, once the PEG is off, then it's off. The cells can adhere, and you cannot go back in the end. This can also be achieved by other molecules, of course, but I mean, switching cells to adhesion and non-adhesion is sort of a really boring thing. A very interesting thing that happens in biology is that when you try to de-adhere cells, that cells often just don't get off a surface, but that they enhance their adhesion. It's a little bit like a mountaineer hiking, trying to climb up a rock. You start to pull at his or her back, and then adhesion is increased. Of course, because you try to hold on to the rock. And how does this now look like for a cell? So initially the cell looks like this here. So it's 0 seconds. It's just the start of the experiment. And then there is a microneedle, a glass microneedle that is used to shear the cell. So initially it's just at the side of the cell. And then someone just used a micromotor to apply a shear stress to the cell. And you see that here the cell went off the surface, so it was de-adhered. But here, this area is still sticking to the surface. And in this area we are now looking at, and this area initially looked like this. There are white dots, and these white dots are adhesion proteins. So the cells stick to the surface by adhesion proteins. We can mark them with fluorescence microscopy, and they don't just stick to the surface by an equal even distribution of these adhesion proteins, but they form clusters. This is a very basic principle in adhesion science. You also find it in other systems, like even in animals, that a splitted contact makes sense, because then you have to apply the detachment force to each of the contacts when you want to remove it, and you just not remove the whole thing immediately if one contact is broken. So the cell really has these distinct adhesion spots that it adheres with, and we can see them in fluorescence microscopy here. And now when a shear stress is applied, you can see that these spots increase their size. And this happens due to the stress applied by the needle. I mean, it's a complex phenomenon behind it. It has a lot to do with you apply a force to proteins, they start to unfold, they open new binding sites for further molecules. And this is approximately the principle of how such a growth works. So the cluster really consists of hundreds of proteins, and they are all enriched when this force is applied from the external side. So that means cells have a dynamic mechanism to adapt their force. And of course, they can also release adhesion. Yeah, it's very important that they can dynamically steer their adhesion and their stickiness. But the main message from this graph was really cells react to external forces. And we wanted to understand what happens during this growth of the adhesion sites by making substrates that really pull on the molecular basis of cells. So these molecules consist of a head group that can bind to proteins of the cells, bind to the integrins, and they have a photo-switchable group that is not only switching just from a trans to a cis state, but it can oscillate. It's a so-called push-pull isobenzene molecule. And we put this oscillating handle to the surface, to a glass surface. And when you now shone visible light onto the substrate, then the RGD molecule was starting to oscillate. You could stop it again. We could start again. You can really repeat this for a long time because the molecules are very, very photostable. So it's repeated frequently. Our idea was that we wanted to see what happens if you really pull on the cell with molecular handles and not just by using a huge microneedle. And we also wanted to see at which timescales it happened. I mean, in the slide before, we had seen that the timescale that they had with a glass microneedle was at the scale of a few minutes. But biological processes often happen at the timescale of a few seconds. So we wanted to explore this in detail. So in the end, you have to imagine these are your handles onto the proteins. It's not nanoparticles here. So the black spot here is simply only this RGD head group that is meant. And they pull on the cells that are attached to these black heads there. In real life, you can really use it in cell culture. So this is just a cell culture flask, and you can use visible light to switch the substrates. Now, as I said before, we are physicists and we often use physical methods to investigate cell adhesion. So I'm sure that all of you know atomic force microscopy as a method from material science and physics to characterize surfaces in topography and also mechanics. But we use atomic force microscopy to do single-cell force spectroscopy. And in single-cell force spectroscopy, we have a cell attaching to the cantilever. It's a tipless cantilever because we don't want to harm the cells. We want to keep the cell happy, so that's why the cell is smiling here. And we press the cell onto the surface. We measure the deformation of the cantilever and we measure the forces associated with the detachment of the cell in the end. And we not only can measure the force that is needed to initiate the detachment, we can also see, we'll see it in a second, we can also see the single ruptures that are associated with the detachment in the AFM curve. So this is a very interesting tool to really study cell detachment in detail. There are a few parameters that we can read from that. I mean, it's partly very similar to what you do when you investigate materials. For example, this testing of material elasticity when the cantilever approaches the surface and you can measure how strongly it is bent upon a certain indentation depth is very typical for soft materials. So we can read out the elasticity of the cells from that. Then we can measure upon detachment the maximum force that the cell holds to the surface. So this is the force that we call detachment force, at which the detachment process is initiated. We see single rupture events, and a very important rupture event for us is the final rupture event, because these single rupture events are actually very hard to analyze. I mean, you must imagine you have a large variety and a large number of connections of the cell to the surface, and then one of them ruptures. So the question is, what do you really measure? So for the final rupture event, we assume that only one bond or maybe a few ones rupture at the same time. So this is a much more reliable parameter in the end. When we now look at the data, how they look like, when we let these connecting molecules oscillate, we saw a very, very interesting effect that we had not expected like this. So our contact between the cell and the surface was just 1 second. So it was way faster and shorter than the experiment done with the fluorescent molecules where the cell was sheared with a glass microneedle. And if we had this 1 second of cell surface contact and analyzed the cell detachment force as a function of light off and light on, where light off means the molecules are not oscillating and light on means that the molecules are oscillating and thus pulling on our integrin proteins in the cell, we see that the cell detachment forces is really higher in the case where the molecules are oscillating and are pulling on our cell proteins. The effect was even more pronounced for a contact time of 3 seconds. So you can clearly see that the cell detachment force was enhanced. So our interpretation was that the reinforcement that we saw with this glass microneedle as well already happens at very, very short timescales. It could just not be recorded in the case of fluorescent molecules because probably the change was too small. And it was an evidence or another indication for that you really have a reinforcement, and now even with molecular oscillations. We also did a detailed study of this final rupture event because we wanted to know if even the single molecular bonds are changed. And indeed, we saw a tiny, tiny shift of our last rupture force, which is here on the x-axis, towards higher forces when the light was switched on, so when the molecules were oscillating and applying a force. So here to the y, on the y-axis you see the relative frequency, on the x-axis you see the last rupture force. And also here the effect was more pronounced at a higher contact time. So also single ruptures were reinforced by molecular tickling. In the end, we also did a biological study where we checked the protein expression with and without molecular tickling. And we also found that adhesion proteins and proteins involved in the adhesion of cells are expressed more when the cell is mechanically stimulated. So this was the first evidence that a stimulation of cell adhesion at the molecular level is possible, and we are now going to the next steps to really explore this into detail. So with that, I would like to move on to microstructured biomaterials. So cells are in nature often present in 3D. So this is an example of cells in a collagen matrix. You see here some fibroblast cells, and you see the fibers of the collagen matrix. With this figure, it's also clear that the cells, when they move, really have to be able to migrate through tiny constrictions. So they must apply forces to their surroundings, they must be able to squeeze through, and this is actually particularly true for cancer cells, for infectious diseases, where cells have to crawl through dense tissue to reach new host tissue, to in the end damage new host tissue. There are, of course, a lot of biochemical processes involved, but there are also structural processes involved. So to study this in detail, we made materials that contain microchannels, and actually microchannels, introducing them in a very small diameter configuration into materials is not too easy. I mean, what you see here is our result. I will explain in a minute how it works. You see channels that have a size of about 10 micrometers, so the average was like between 5 to 15 micrometers. And this is something where it's very hard to reach with 3D printing still. Therefore, we have chosen a different strategy. We have chosen zinc oxide tetrapods that can be very easily made from zinc microparticles by a flame transport synthesis. And these tetrapods were compressed into a small tablet and sintered together so that really the connections between the zinc oxide tetrapods were very firm. Then a hydrogel was added on top of these tablets, so they are really at the centimeter scale. I mean, in contrast to the best 3D printers, really at a very, very, very big scale. And the hydrogel was added and polymerized. Then the zinc oxide was removed by a mild acid treatment. Then you can swell the samples because, of course, hydrogels take up a lot of liquid, and then you can functionalize if you need it. And this procedure is compatible with a lot of materials now. It works with polyacrylamide, with Gelma, also with thermoresponsive materials, and also with PEG-DOPA or other polyethylene glycol materials. So in the end, in this way, we can get materials where we where we have tiny constrictions in order to test how the cells migrate through such a material. So how does cell migration now indeed look like? So this is an acanthamoeba that migrates through such microchannels. Acanthamoeba are unicellular organisms. They live in ponds, they live in tap water, they live in lakes. They also like to live in swimming pools, and sometimes you can read in the news that there are outbreaks among contact lens users who didn't clean their lenses properly. And what these guys then do is they simply destroy your cornea. So it doesn't look very nice. It's a pretty ugly disease and it goes very fast. So within 1 day or so, the complete cornea is destructed in these cases. It's also very hard to treat disease because if you use conventional drugs, then these organisms are able to encyst into a double membrane system. So they really escape from the treatment, they go into the cyst state, and they can then reappear after the end of the treatment. And what we were now interested in is if we are able to capture such acanthamoeba from solution, because it is known that acanthamoeba also in the soil have to walk through very dense sand areas and soil areas. And indeed, what you can see here, first of all, is such an acanthamoeba. that during these 4 minutes crawls through the channels. I mean, the video jumps back all the time, but you can see that it really reaches out into this channel, but it somehow gets stuck because it carries a lot of cargo. So getting stuck is a very important thing when you think about making channels into materials, because of course you can change cells by that. And what we interestingly found is that when we had the acanthamoeba in our incubation medium, and when we had a tablet with such a polyacrylamide sample that contained microchannels in there, then we found that after 120 hours, there were many more acanthamoeba left in the solution when the material did not have microchannels, compared to the situation where the material contained microchannels, simply because many of the acanthamoeba went into the material and got stuck there. And this getting stuck, we can also record quantitatively. So this is a sort of a busy diagram. What you can see here is the average speed of the acanthamoeba and their diameter. So we first of all checked, does the diameter of the channel, which in the end then tells you what is the diameter of the cell, how does this influence the speed of the cells? And these black dots here show you, well, the average speed inside the channel is not really strongly influenced by the diameter of the channel. Below 7 micrometers, we did not find any cells simply because they did not fit through. I mean, they have a very compact nucleus, and they did not walk and migrate through thinner channels. But compared to that, on a bulk material, just a 2D surface without any constrictions, the cells could be much faster. So we could really control the average speed. The important thing is now when we look at the bulk situation, which is the blue one here, all of the cells are migrating. In our materials, if we look in the channels, less than 50% are migrating. A lot of them get stuck. This is probably a result of the construction of the material, because there are larger cavities at the center of these tetrapods. I mean, the materials were made by tetrapods fused together, and at the center of these tetrapods, where the side arms go out, there's a larger, in the end, a larger cavity in the hydrogels. And we found that the acanthamoeba tend to enrich there and stay there, and this is their resting place. So we can indeed control cell migration by providing 3D structures. We can also do something else. Let's go back to the microchannel geometry. This is the tetrapod shape, and in the end, we get a certain structure in our tetrapods. We embed this tetrapods, these zinc oxide tetrapods, in hydrogels. So our hydrogel in the end has these channels, but everyone working with hydrogels knows that you can control the stiffness and also the water content of hydrogels by varying the crosslinker concentration. And for our polyacrylamide materials, we varied the crosslinker concentration between 1 and 16 volume percent. And in this way, we could also change the Young's modulus. And we saw a slight change of the diameter of our channel as well. It was just a very tiny change, but I think you agree that for the stiffer materials, which are stronger cross-linked, of course, the swelling after the fabrication procedure is less, and therefore the channel diameters are a bit smaller. And now this is super busy, and you should not get confused here. The real message is we can put cells, also cells from the human, now not any infectious amoeba or really just mammalian cells, onto these materials, and then we can check where they are located. Yeah, so they can either stay on the sample surface because they cannot enter the channels, they might stay in a channel, but they also might stay in channel intersections or in these larger cavities. And we really did a super detailed study on that as a function of stiffness of the material. And we found that, well, the channel was preferred in the very soft samples, and it was less preferred in the really hard samples. But in the hard samples, the cells tried to stay outside. We could also see that the nucleus sometimes ruptures. So in phase contrast, you almost don't see anything. And this nuclear localization sequence you can see outside of the main nucleus. So you can see here the main nucleus of the cell. This is a histone sequence. This H2B RFP is a stained part of the nucleus. And when the nucleus is damaged, which can happen when the cell moves through a very, very thin channel, then you see that this nuclear localization sequence leaks out into the rest of the cytoplasm, and then you can see something like this here. So we did a lot of analysis, and we were a lot interested in how is cell positioning influenced, how is nuclear stability influenced, and we saw a strong impact of location of the cells as a function of stiffness. However, I mean, the location of the cell, how to detect it is always tricky. And I mean, in these experiments that we have done there, it was still pretty easy because we looked at single cells, we looked in confocal microscopy, and we just looked at very, very thin samples. The whole story becomes really complicated when you go to thick samples and when you really want to see where large cell ensembles are located. And this is now where a magnetic method comes into play. Because what you normally do is when you want to locate cells in large 3D materials is you fix the cells. That means you fix their state, but you kill them at the same time. So you take out all the dynamics. And then you make sections of your sample, and then you go to microscopy or, in the end, you go to microscopy, but you can go to different types of microscopy. So you really have a very tedious work in analyzing all these slices, and you cannot just continue to do this experiment with the same cells because you fixed the sample and the experiment is stopped and you have to redo the whole thing. And therefore, we had the idea to check the location of cells by magnetically marking the cells and trying to find out their position by checking the magnetic signal. This can be done by magnetic resonance imaging, also by magnetic particle imaging, so really complicated, expensive methods. And we try to find a different method which is based on delta E effect sensors. So for those of you who are more in the biomaterials field and less in the magnetics field, maybe you just skip this part and come back in a few slides. But I hope that those of you who are more into the magnetism field get really interested. So in Kiel, we have a collaborative research center, which is a big research collaboration of like 15 to 20 groups. And there are a lot of experts on making sensors that combine a magnetostrictive layer with a piezoelectric layer. And the interesting point here is that when you excite the mechanical resonator that you have here, the magnetic field that is applied to the magnetostrictive layer changes the Young's modulus of this whole cantilever. And in this way, you get a change in the resonance frequency as a function of the magnetic field. So this means by measuring the change in resonance frequency, you can find out about how big the magnetic field is that has been applied. And now these sensors have a lot of tricky aspects. I just want to go to the rough details. The main trick of the sensor is, I mean, you see here the resonance frequency to the left and in the x-axis the field. And normally the dashed line is the typical characteristics of this sensor. And these sensors, they have very good operating points for measuring magnetic fields, of course, in the linear region. Yeah, but these linear regions are naturally not around 0 Tesla, because here at 0 Tesla they have a maximum of the resonance frequency. But what you can do is you can apply magnetic bias field, and in this way shift shift the curve, so that you have this ideal operating point with this high slope here. So that means a high magnetic sensitivity around 0 field. And that means if you have small fields on the sample, you will get a large change in the resonance frequency. So you need an offset field by a permanent magnet. And this is what the principle that the magnetic susceptibility in particle mapping is based on. So there is a permanent magnet that provides the offset field for the sensor, and at the same time excites particles that in the end are in our cells. The cells are marked with magnetic particles. And then the sensor will be able to record the field from these particles that are in the cells. So how does it look like? We had a very, very simple setup, but it's actually running perfectly. So it's just a CD. And here's the permanent magnet. The sensor is actually at the bottom, below the CD. And the sample is rotating through this gap between the magnet and the sensor. So it's exactly what you saw before. Yeah, you have the magnet here on top. This excites the sample and provides the bias for the sensor. So the magnet really has this dual function. And this is just a setup that we then see later on. And by applying a rotation on the sample, you can do all this at the right frequency to really have so that you're very close to the operating point of the magnet. And you, of course, also have a very high amount of data, so that in the end we could nicely record the sample in here. So this is just a projection of the CD. And you can see in the color code the magnetic field that was detection, and X and Y are just X and Y on the CD. And this was indeed the sample that we could see. The signal that was detected, so the signal you can see here, this is the field. This is in the end the signal that we get for the field at a certain position in angular coordinates. And when you look at the maximum that we can get there, we find a linear relationship between the field and the iron content. So Niels did a very detailed study on checking different concentrations of this iron content in these tubes, and it was very clear that there is a linear relation, so we could really use it to calibrate. We also did a spatial resolution analysis here with macroscopic samples having a lot of magnetic particles at different spacings. Oops. And we could see that we could still distinguish them at a position of 4 millimeters. I mean, of course, this is not single cell resolution, but if we think about applications of materials in tissue engineering, we are indeed at the scale of a few centimeters. So simply a resolution of having, after some improvements, hopefully with a currently running PhD thesis, getting at some point of 1 millimeter resolution, that would be perfect. And I think it's very realistic. We have also been able to do first cell experiments with such a system. So this is a hydrogel, an alginate hydrogel, where a lot of magnetically labeled cells were in. So you can see here some cells that were inside this material. They also contained a lot of magnetic particles. And in the end, we got a signal that could be distinguished from the noise and show that our cells are located here in our dish. So this was the first proof that we can indeed see cells embedded in a gel and marked magnetically, and we are now striving forward to really improve the system such that we can get a millimeter resolution, maybe also going to 3D experiments, because currently everything is just in 2D. So the big impact would really be getting dynamic cell imaging, magnetic cell imaging inside hydrogel scaffolds. So with that, I would like to extend very similar strategies as the one that I've shown you before towards the regime of actuators. So why that? It's not truly a biomaterial, but it comes very close. So just think about how you grab a pen. I mean, when you transfer a pen to a different person, you have a very gentle grip. This is also important for having a safe work environment in the end, because if you interact with something that is hard, it can also damage you. So that's why soft robots have become a very, very important strategy for making new robots, particularly when they interact with humans. But of course, soft robots are also interesting when you want to think about body part replacement, if you want to have a human-like touch, but also when you think about processes happening at the nanometer and micrometer scale, because often there you have very sensitive objects that can be damaged. Current strategies in soft robotics often employ exoskeletons. So there has been a lot of improvement on really just having these gloves that give you back your gripping ability when you have problems with doing that. Then there have been very clever gripping devices by pumping up silicone actuators, but the main mechanism are electrical motors. You have a lot of cables that you have to transfer. So it's maybe not the perfect strategy on the long run. And certainly it would become very, very hard when you go to tiny scales. Just think about what you need for making an electrical connection at the micrometer scale. And one polymer that is very attractive in such soft robotic devices is Polynipum. Polynipum is a thermoresponsive polymer that is below 32 degrees in a fully hydrated state. So it's a hydrogel containing a lot of water. Of course, when you give it the chance to have water. And above 32 degrees, it is in a dehydrated state. So the water all goes out from this material. So this phase transition is very, very essential, and as it's already sketched here, in the fully hydrated state, this material has a large volume, whereas in the dehydrated state, it does not contain a lot of water and it shrinks. So the interaction between the polymer chains is here dominant, whereas here the interaction with the water is dominant. And of course, you can nicely use this for robotic systems, because now you can achieve a volume change by a change in temperature. And this is a pretty cool video that a guest student from Canada and my PhD student Tobias Spratte have done last fall. So it is it's just, yeah, it looks like a worm, yeah, but it was made of polynipum of different concentrations. So the top layer, where you see my cursor now, that was made from pretty much cross-linked polynipum. The bottom layer was made up of less cross-linked polynipum, so it contained a lot of water, but it could also press out more water. So it would shrink more when the material is heated. And they had a small heater here at the bottom. And what you could see, because of the ratchet architecture here of the surface, this walker, now it's going to the cool state again and taking up the water. Then it's heating again and shrinking. It can walk on this ratchet surface. So I will just restart again because it's such a nice demonstration here. So it shrinks, and the inner part here shrinks more than the outer part. So that's why the bilayer is needed. And it cools down again and relaxes. And then it's heated up again and shrinks. And in this way, it can just walk forward. And this can now also be transferred to smaller dimensions. But what we observed, so please first look at the left-hand side. So what we observed was when we used bulk Polynipum, as you just polymerize it in a standard and also as it was done for the Walker, it has a volume change of only about 12%, and the Walker could only be achieved because of this trick that there was a bilayer. Also, the shrinkage time was pretty high. That means the process is very slow. And also, of course, the speed of this Walker was very sped up in the video that I showed you. So with such a volume change, I mean, you cannot generate a really cool actuator in the end. But we applied to the Polynipum the same strategy as we had applied before to other hydrogels by introducing microchannels. And the microchannels are now important to transport the water out, because the whole transition from a large material to a shrunk material is based on getting the water in and out. And in this way, the time of shrinkage could be could be shortened to 60 seconds, and a volume change of 82% could achieved with this strategy. So this was pretty amazing. We could also observe very nicely that there are indeed channels here inside the material. So these are SEM images of specifically stained and fixed hydrogels. So you can nicely see that we have channels. So it's not just that we think there are channels, they are indeed stable and stay there in the material. And when we analyze actuation efficiency, we see a clear improvement by channels. And I really want to guide you through these busy slides here. So the black dots, this is Polynipum without microchannels. So no microengineering, just the bulk one, just synthesized. And what you can see is here is the weight on the y-axis, and this is the time on the x-axis. The black dots show the volume, show the weight, so the swelling, the deswelling when you heat up. And you see that for these black dots, where there are no microchannels, well, the weight does not change so much, so the shrinkage is not so much. In comparison, when you have channels in there, we can clearly see that there is an extreme change in the weight. So a lot of water is pushed out within a very, very short time of just 10 minutes. On the other hand, when you cool down again, so let the water go in again, well, I mean, the bulk Polynipum without microchannels, yes, it relaxes also back almost to the original shape. And for the microchannels, this also happens, but it all happens a bit slow, yeah, because probably also the channels are glued together a little bit. So the swelling is not so easy to explain and to describe, but the shrinkage is really extreme for the microengineered Polynipum compared to the bulk Polynipum. The effect is extremely visible if you look at the channel density, or if you look in another representation where we just really look at the normalized weight as a function of channel density. If we have 0 channel density, that means just a bulk material. We have this very, very flat curve. The material almost does not lose any weight, but it loses a lot of weight the more channels are inside the structure. And the cool thing now was that it was possible to use this material for a thermoresponsive gripper. So there is a 3D printed frame here. The material is still at 22 degrees. In this case, in this situation, you see the material is hydrated. It's a hydrogel. It's pretty large. And we will now try to grab such a small sponge particle. And when I start now here, this was cycle 1. And you have to stop. So you see at high temperature, the material really becomes this whitish dehydrated material that is very much reduced in volume, and it can in this situation grab this sponge, and you can nicely repeat it. Of course, it's not that fast, yeah, but if you imagine underwater applications or applications that don't live from having a very, very high speed, this can indeed be a very nice application of the strategy, and it would not be able to do this with just a bulk Polynipum. You really need this micro-engineered Polynipum to get the force to hold on to this sponge. So that means putting channels into hydrogels not only makes sense for biomaterials, but also for very different systems, for example, in soft robotics. But of course, it can also be interesting for other systems. Just think of drug release or storing something into the hydrogel that should get out later. So with that, I would like to conclude, and I would like to advertise a conference that is happening in fall this year. So it's on molecular systems engineering for bioapplications. You find the information on our website, and we are, of course, very happy to get your applications on this conference. The conference will happen in the city center of Heidelberg, approximately here in this area in the Heidelberger Akademie der Wissenschaften, which is a very, very old scientific institution in the city, and it's organized by 2 of my group members and a colleague from the organic chemistry. And last but not least, I would like to thank my team. So you can see we are a very diverse team. We have many, many different backgrounds, and we have very nice collaborators at different places. And of course, I would also like to thank our funding agencies who have really followed us over the years. And with that, I would like to thank you for your attention.
Very good. Thanks so much, Christine. Very, very nice talk. Actually, it was, I don't know, a combination of different areas, and the work that you are doing with the other groups, it looks that it is very multidisciplinary, and that you are succeeding in that work. I don't know if you can comment on that. How do you manage to put all the people under the same hat to work together on this?
That's a funny question. I think we just do it, yeah. Well, I mean, I have been trained like this all the time because I did my PhD already in a group that was very, very interdisciplinary. And I think you need both. Yeah, you need people who are very disciplinary because sometimes you just need to be able to ask them, but you also need to have people who can speak the language of the others. And I think we are more people who are able to talk to many different fields and then also pick up the information that we have. And I think here on campus, we really have the perfect environment because, it's a big campus and we have many colleagues that are very open for collaborations. And I think it's a bit of the spirit of Heidelberg University. So also teaching here has been very interdisciplinary for many years. There are now many interdisciplinary programs, and I can just motivate you and your colleagues. So if you have students who are interested also in international master programs, we even have an international master plus PhD combined program now. So there are a lot of opportunities.
Yeah, great. Here we have the first question. It says, Eduardo Leiva, thank you very much for your talk. Very interesting. Have you carried out studies of the rheology of your systems? Does rheology play an important role in the design of your systems?
Yes, rheology plays an important role.
Mm-hmm.
I have rather talked about stiffness because we have most experience now with stiffness experiments, simply because traditionally cellular systems are known to react on substrate stiffness. So this is where the story started. So we have started to do indentation and so on, but now it's pretty clear that also viscous properties matter. And particularly in the past 2 years, we have started to do rheology. We have not done it on these systems now that I showed you with a microchannel containing Polynipum, for example, because there we have mainly been interested in, which pushing force can you achieve by the actuator, which stiffness do you have? But we have started to do it, and I think it's a topic that comes in more and more because people start to realize it's not only the elasticity, it's also the viscoelasticity. So this is really an important point.
Great. And here I have a couple of questions. The work that you were performing with aerogel and with these zinc oxide tetrapods, I think it was a very nice collaboration, maybe with the group of Raina Adelung, no?
Yes.
Yeah, you come with nice solutions for different stuff, and with these tetrapods, they are putting tetrapods everywhere, but with very nice solutions for everything. And here I'm just wondering, you were making some test putting this acanthamoeba, and this amoeba were coming inside that tubes left by the zinc oxide tetrapods that they're not anymore, no? But what I was just wondering is also the aerogel has also some porosity, no? That
Yes.
sucks some water, or the solution that you have there. Are the amoeba not coming to these parts of the aerogel?
The pores are too small, yeah,
Okay.
but the pores are important. So don't start to
Yeah.
do these experiments with silicones, for example. The cells will just stop. I mean, we
Mhm.
once had these acanthamoeba in tiny channels because, well, you know, from the biophysics part of thing, I mean, it doesn't make sense to go to 3D. Yeah, you would like to have a 2D channel and just see how the cells move through. But the point is you need the hydrogel because in that way you have a nutrient transport. So these cells, of course, need a lot of energy and a lot of nutrients. Yeah, just think of sugar to be eaten, yeah, to move forward. Yeah, and if you don't provide this, for example, in a thin channel where you only have diffusion then from the front and from the back, that's not sufficient. So we were extremely lucky that this hydrogel system that we tried really allowed the cells to move from the very beginning. And it's also about the metabolic agents. Yeah, so the cells, of course, I mean, they have a metabolism. So they not only take up sugar, they also leave stuff behind. And if this cannot be transported away or is diffusing away, of course, things change a lot. And this is probably even one reason why 2D and 3D materials are so different with regards to cell. I mean, cells that are cultured in 2D and in 3D are different in the end. We know that. And part of it is certainly because they are in a different chemical environment in the end, because in 3D they might have less nutrients, but they have more of the metabolic agents that they produced around. So this can influence them in the end.
Yeah, nice. And here I was also wondering about if the pores left by the tetrapods are linked all together like
Yeah.
a tube, or that's why the amoeba is coming just at the end of this link and is staying there.
It's a very, I mean, I see that you know the system, yeah, because I mean, for the others, you have these tetrapods, yeah, okay, here's the camera, you have these 4 legs, and then the next one is somewhere coming there. So this means now, here it is. So this part of my finger, yeah, that's a dead end for a cell. And I think that's indeed the point that could make trouble. We are not sure. But that's also why we are switching away from the zinc oxide right now, because we have these dead ends where we don't know for more biological applications what's actually happening. And also the tetrapods, I mean, they are not absolutely straight. Yeah, they are relatively straight, but it's more like this. Yeah, so either the cell walks towards smaller diameters or towards larger diameters. And I mean, this was simply very hard to achieve. And I also must say, of course, we can now make with direct laser writing extremely tiny, nice structures. But of course, we cannot reach the centimeter scale. So the zinc oxide still is perfect if you want to go to large materials and want to have large scale biological interactions, and in the end also statistics matters. Yeah, so we go now a lot to the tiny structures that are very, very precise, but it is indeed yeah, I mean, these tablets still have the advantage at the large scale, yeah. And sometimes the detail, I mean, it could be that the dead ends don't matter, yeah, that they just level out over time.
Yeah, nice. Thank you for the answer. Here's again Eduardo Leiva. He's asking, do you consider that the bulk rheology macro scale is correlated with the rheology at the micro and nanoscale?
Well, yeah, there is certainly a correlation. We have not analyzed it yet. So we are currently rather going for the macro scale rheology. We have just received a system where we can also go for the micro scale. It's certainly an interesting question because, I mean, at the micrometer scale or nanoscale, you have a lot of impact from the water, yeah, and you really see the effect very, very strongly.
Yeah, thank you. And now that you were speaking about these materials that is complicated to measure or to track the cells in a 3D material, and that you are performing some experiments in 2D with the magnetic sensor nowadays. I was just wondering also about the working principle you explained already, but also about the 2D 2D map that we get there. Actually, the sensor is also moving in the radial direction, just to get also information not just from passing the disk, but also from inside the disk or something like that. Or do you have an array of sensors below or how does it work?
So the cool thing was we didn't move the sensor for these
Okay.
experiments. Yeah, so this was really just a recording from this one position. But this is exactly the next step, yeah, to vary the position and then check how does this influence to vary the position really in all three directions of space. And yeah, arrays is an issue. Yeah, so it's really it's a plan, but also with arrays you have to be a bit careful because, as my colleagues told me, you know, I'm really not the expert on the electrical engineering, but what can happen easily when you have arrays, that also the noise sums up, and therefore when this is the really the tricky part that we cannot handle. So there people are often thinking about micro arrays of hundreds of these sensors, but if you cannot make the electrical connections and the readout connections such that you really minimize the noise, then you have not won anything. Yeah, so that's why. But I think for the very simple setup that we have set up there, it's already pretty cool, and I think the next steps will be very exciting.
Yeah, actually, it's already very exciting, this measurement, and the resolution is not bad. But I was also wondering if this resolution is restricted by the size of the sensor that you are using and producing there in Kiel, or this cantilever that you are using is limited by this limits the resolution of the map that you get there.
To some extent, certainly. Yeah. So yeah, but probably it is very hard to really change the size and get enough high signal then still, compared to having a few sensors at different positions.
Because
and there are really applications where it might make sense to just have this resolution. You know, I mean, I just heard a talk about tumor margin detection, yeah, and it still takes a long time. You know, when you have a tumor surgery, they send then the tumor to the pathology, then they look in light microscopy. It would be just cool to have a device, you put the magnetic particles on, that they stain the tumor cells, and you can just go over it very quickly directly next to the surgeon. You don't need extra experience. You can just say, well, okay, tumor is completely removed. And I think these are applications where something like millimeter resolution are absolutely sufficient. The sensitivity should, of course, be high. That's what we are working on. But it's certainly not that we will reach single cell resolution. But I think bridging the gap between the super high resolution methods that are extremely expensive and having to fix, to cut, or to stain the cells, this could be a very nice complementary method.
Actually, it's already impressive that this measurement that you are performing there, and also the sensitivity of some nano Tesla that you have there is really great. What you have,
Yeah,
no,
it's great, and I hope that your previous student Angeles de la Cruz will be able to improve it even further. So I assume that she's listening. So I think we will make very nice progress very soon. Yeah.
yeah, very very nice. And also, I don't know if you can comment a little bit at the end of your talk when you were saying thanks for different grants and so. You were, and also at the beginning, you were talking about this center that you have, collaborative center with Kiel and so, and the different areas. That's why I was asking, how do you manage to put everybody under the same hat? Uh, if you could comment a little bit about that project of different groups, this collaborative center.
Yeah, in this collaborative center, I mean, there are big groups in material science who really work on ME sensor fabrication in the end, so this magnetoelectric sensor fabrication. They improve also the clean room technologies. They also take care of the electrical readout of all the sensor characteristics. And then there are, of course, several applications for this. So I'm more on the application side. And there are people from electrical engineering together with medical scientists who are, for example, really using the sensors now for motion recording. Yeah, I mean, elderly people, they often have problems to walk or have some shakings and some motoric diseases. And they are now, for example, building jackets with magnets and then having the sensors in other places of the bodies and doing long-term recordings, because the recordings are currently mainly done then by video analysis. And this is something that you cannot do all the time. But whereas with these jackets, where you have a continuous readout over days, you can really get some data. Yeah? And this is one idea. And other directions really go into sensing of brain currents and also of cardiac signals in the magnetic fields. Yeah, I mean, yeah, if you have electric signals somewhere, you also have magnetic signals. So that means you can record magnetic signals from the heart and from the brain, but the heart is much stronger in the magnetic field. And they have also built now a special mattress with embedded sensors to record an MEG just when the patient is lying on top, or that really the patient can be recorded overnight without needing EEG electrodes. Yeah, putting EEG electrodes, I mean, that's a nasty thing. That's something that you would not do as a patient every night at home, but just lying on the mattress where you're anyway sleeping. I mean, it's no issue if in parallel your heart is monitored. So that's why there are a lot of activities going in this direction.
Yeah, thank you. It is, yeah, actually, your work that you presented today and all this, what you are mentioning, it is very encouraging to follow some of this stuff or research. Actually, there are like 20 people following us on YouTube now, and some of them are students also from material science and also from electronics and yeah, different areas and some bio applications. No? And I think all of them are they got something from you today, and also you invited us to some collaboration also with Heidelberg, and also that you have a nice master program there, no? And also yeah, you know you have also PhD, not as a program maybe, but yeah, you have several students.
There are regular opportunities, yeah, not only for me but also for others. So just check out, and I don't know if some people at your place know Angeles, yeah, but I'm sure she's happy to help. So you will find her on my webpage, and I'm sure that she can maybe give you the link between the German system and the Mexican system. And she also came to Heidelberg as an internship, or not to Heidelberg, at that time still to Kiel as an internship student. So this is something that we could, of course, follow for collaborations also.
Yeah, thank you very much. And maybe a last question from my side. I'm asking a lot here.
No worries.
So from the first part that you were presenting on the photosensitive cells or something like that, no?
Molecules on the surface, yeah.
That you were kind of scratching with the AFM, the microscope with the tip. Were those measurements done in contact mode?
We don't use any of these classical modes. Yeah, we have a tipless cantilever, so we don't scratch the cells. We really glue the cell to a flat cantilever, and then we don't use any of these classical modes. It's just for spectroscopy. So it's really we also have an AFM head that even cannot move in X or Y. It can just go up and down.
That is.
Yeah.
I was going to ask if you were not actually. What we saw there is kind of some oscillations, no, from the cells in the results that the corpus.
Oh, we saw that the cell is pressed to the surface, and then how it's ruptured and how the different rupture sites
the
go
frequency
up. Yes.
or these characteristic frequencies were coming from the cell, no, or from.
Yes. You saw the noise. The noise is thermal noise a lot.
But also, the results, the graphics that you presented are in the y-axis. It was like frequency. I'm not so pretty sure.
Sorry. That this was the number of ruptures. We just normalized it, and then in our field you call it frequency. I'm really sorry for this misunderstanding. So it meant, when I apply a force, then the last ruptures are shifted to higher forces, and the relative frequency when this occurs was plotted to the y-axis. In the end, it was just the normalized number of ruptures that we observed. It was just a histogram. Yeah.
Okay. Yeah. No, very good. Yeah, I was up to ask if it has some relation with the frequency of oscillation of that tapping mode or something like that. But
that's what we are exploring now.
Very
will
good.
take some time.
Very good. So I think we could finish here. And thank you very much, Christine. I hope we will
Thanks
see
for the invitation.
In another conference, maybe here or there in Germany. Now the times are a little bit strange, but nevertheless we are still moving, no?
Exactly.
And I hope we will move again to hug each other somewhere here or there.
Exactly. Yeah. Okay. Thanks for the invitation. Thanks to the audience. Bye bye.
Thank you very much. Ciao, ciao. See you next.
Bye.